Good morning student. Today I am going to explain you electrical synapse. In our previous lecture we have discussed about the structure of the neuron. Then we have studied about the chemical synapses. Today I am going to explain you the electrical synapses which are occurring. So what is an electrical synapse? In electrical synapse, there is a gap junction between the plasma membrane of two cells. In chemical synapses, we have studied that the presynaptic membrane, postsynaptic membrane, and there was a gap between it that is called as a synaptic cleft. But in electric synapse, the two plasma membrane of the cells means presynaptic and postsynaptic neurons, they are in contact and there is a gap junction between them. So gap is also there and junction is there. So gap junction, it is composed of 100 intracellular channels called connexon. Now this gap junction, it is made up of connexon. What are connexon? Connexon is composed of protein called as connexons. This connexon, it is made up of protein which are called as connexons. So this question is asked, what are connexons? They are joining the cytosols of the two cells. Means the fluid which is present between the two cells, inside cells, it is joined by this protein channel and they are forming a tunnel. And what is the width? That is 2 nm. So in electrical synapse, you can see there is a gap junction between two cells, means they are in direct contact. Now as you see in the diagram here, it is shown how in electrical synapse the gap junction is there. You can see in chemical synapses what was happening, the impulse was coming, then it is coming to the terminal of the axon, then this calcium gated channel, they open, influx of the channel of influx of calcium inside. So synaptic vesicles, they diffuse, come to the presynaptic membrane, and here what they are releasing the chemicals. This neurotransmitters, it is received by the receptors of the postsynaptic membrane. So a long procedure is there and it is taking time. But in electrical synapse, here you can see what there is a bridge gap junction between presynaptic and postsynaptic cell. This very well, it's marked by an arrow and you can see. And the space between the pre and the postsynaptic cell, it is 3.5 nm to 20 nm. So the space is from 3.5 nm to 20 nm. So this is the normal cell. Here you can see very well. This is the anatomy of the electrical synapse. Now coming, as I have explained you, the connexon, they are made, what are connexons? They are the proteins which are there in the bridges. So it's very well clear in the diagram. You can see the cytoplasm of the two cells, that is the cytosol. It is in contact by these connexons and they are forming a tunnel of it. And what are these connexons? They are proteins. So these rounded structures, green you can see, they are the connexon and they are joining. And here these channels, ion channels, they are there so that influx of ion could be there. So direct these connexons, they are forming the channel, and here you can see the intercellular spaces between the cell. Here is the plasma membrane of the adjacent cell, ions and small molecules, and channel formed by pores in each membrane. So by pores they are forming the channel in each membrane so that entry is possible. Now coming to the definition, what is an electrical synapse? When neurons, they form electrical synapse, means how? Means you can see in the diagram there is a gap junction between two cells, and so that the ions can flow easily. एकदम तेजी से जाते हैं तो it's very fast. They are only seen in a few locations. If we see example, if you are seeing, then it is in the retina, olfactory bulb of the hippocampus and human nervous system. They are having this electrical synapse and it's very well clear in the diagram also. Now what are the characteristics of electrical synapse? As I have explained you that in chemical synapse they are slow. Why they are slow? Because they are following a long procedure I have explained you when they are releasing their chemicals. These neurotransmitters should be received by the receptors, and then further they are flowing. But in electrical synapse, the cells are in direct contact by a gap junction, so they are fast. Second thing is they are synchronized, means they are organized in a proper way. A presynaptic nerve impulse will always reach the target position in electrical synapse. अब when impulse, suppose कोई impulse आ रही है, chemical synapse में अगर वो आई impulse is coming to the axon, उसकी proper threshold stimulus नहीं है, तो क्या होगा? It will never reach, या तो graded potential में change हो जाएगी वो, but it will not reach the target. Chemical synapse में if it is not of threshold frequency. लेकिन इन इलेक्ट्रिकल साइनेप्स प्रीसाइनेप्टिक नर्व इम्पल्स अगर एक बार इम्पल्स हो गई है, इट विल ऑलवेज रीच टू द टारगेट. सो दिस इज द डिफरेंस. दे आर फास्ट, they are synchronized, and when an impulse is coming, it is going to reach the postsynaptic membrane up to the target. Now you can see this very well, the way I explained you why these chemical synapses they are slow, because as I told you, here you can see impulse is coming, it is coming on the terminal axon. अब ये कैल्शियम से क्या हो रहा है कि जो चैनल्स है ये इनको कर रहा है, इन्फ्लक्स कैल्शियम का हो रहा है. कैल्शियम अंदर आएगा, क्या करेगा? प्री साइनेप्टिक वेसिकल्स साइनेप्टिक वेसिकल्स को प्रेस करेगा. इट विल कम टू द प्री साइनेप्टिक मेम्ब्रेन के अब इनके अंदर जो भी केमिकल भरा है वो उसको निकालना है, जो भरा है वो डालो, send it, and so that these neurotransmitters they come into the synaptic cleft में आएंगे, and here in the postsynaptic membrane they are having the receptors. इनके receptors होंगे ताकि इन neurotransmitters को ले and send the impulse. So it's a long procedure. But in electrical synapse, it's very clear. Action potential is coming here, you can see, and then there is a gap junction between cell. So coupling potential directly it is going. So it's very fast. Now, who is called as the father of neuroscience? Can anyone name? A German pharmacologist, Otto Lavoie, he is called as the father of neuroscience. So father of neuroscience is Otto Lavoie. This question is asked. Why he is called? He got the Nobel Prize in 1936 because he discovered the chemical nature of neurotransmission of which acetylcholine across synapse. Okay, what is the chemical nature of acetylcholine across synapse? He discovered this, and for this he got the Nobel Prize. He is called as the father of neuroscience, Otto Lavoie. Now, if we are coming to the now I am going to explain you about neurotransmitter. This is also very important. The question is asked, what are neurotransmitters and their role? So, what are neurotransmitters? They are endogenous chemicals that transmit signals from a neuron to a target cell across a synapse. What are neurotransmitters? They are coming and then they are sending the signals to the target cell across the synapse. They are called as neurotransmitters. They are very important. Synapses are the junctions between the neurons, and when these chemical neurotransmitters are released, then they are received by the receptors of the postsynaptic nerve, and then it is transferred into the muscles, etc. Some chemicals they are released by neuron. They have little or no direct effect of their own. Means many times the chemicals they are released by the neurons, but they are not affecting. They don't have their own effect, but they can modify the effect of neurotransmitters. So these chemicals are called as neuromodulators. So there is a difference between neurotransmitters and neuromodulators. What are they doing? Some chemicals they are coming and they are modifying the neurotransmitters. They are called as neuromodulator. Very important question. Now, what are the type of neurotransmitters? Neurotransmitters they are of two types. दो तरह के होते हैं. Small molecule neurotransmitter and large molecule neurotransmitters. Neurotransmitters they are of two types. Small molecule neurotransmitter, large molecule neurotransmitters, they are called as neuropeptide. Then there are gaseous neurotransmitters such as the nitric oxide, So coming to the class of neurotransmitters. First, monoamines. If we take the examples such as the dopamine, noradrenaline, adrenaline, histamine, serotonin, they are all these small molecules. Then some amino acids such as the glutamate, GABA, you have heard the name. That is gamma aminobutyric acid, glycine, aspartate, D-serine. They are all these neurotransmitters, peptides such as opioids and endorphins, somatostatin, oxytocin and vasopressin. Some hormones, they are also behaving like neurotransmitters. Now, when we have identified neurotransmitters and neuromodulators, they can be divided into two categories, summarizing small molecule and large molecule. What are the small molecules neurotransmitters? I have explained you monoamines, acetylcholine, it's very important and we have studied a lot about it. Serotonin, histamine. Then coming to this protein, that is the protein catecholamines, that is the dopamines, norepinephrine and epinephrine, some hormone. They are secreted by the adrenal medulla. Then amino acid, glutamate, GABA and glycine. So these are small molecule neurotransmitters. Our question is asked, give example of a small molecule neurotransmitter. So you can give very easily that monoamines, catecholamines, amino acids. Then what are large molecule transmitters? They are also called as neuropeptide. Example, if we take vasopressin and enkephalin, they are the example large molecules. There are also other substances which are thought to release into the synaptic cleft to act either as transmitter or modulator of the synaptic transmission. So in this, there are some purines derivatives like adenosine triphosphate. So ATP and the gaseous nitric oxide, they are also working as a neurotransmitter. ATP is also, that is ADP and AMP and all, they are energy currency as well as the other neurotransmitters. They are working as. Now when there is neurotransmitters, they have receptors. Two type of receptors are there for neurotransmitters. One is the inotropic and second is the ligand gated. Two broad category. So if we take example of suppose of these ligand gated receptors, then if they open immediately when the neurotransmitter is binding, when suppose neurotransmitter is coming on the gate, then they open immediately, ligand gated. They are protein coupled receptors and neurotransmitters binding to a G protein coupled receptors induce the opening or closing of a separate ion channel protein over a period of seconds to minutes, and they are slow neurotransmitters receptors. So in case of ligand, whenever they are receiving this, they open immediately. And so one example is G protein. So they come and they open for a smaller period of time, but they open immediately. Each ligand has many subtypes of receptors, selective effect at different sites. Means at presynaptic receptor, they are also called as autoreceptors and they provide feedback control. तो प्रीसाइनेप्टिक होंगे उनका इफेक्ट अलग होगा, प्रीसाइनेप्टिक पोस्ट साइनेप्टिक रिसेप्टर्स हैं उनका इफेक्ट अलग होगा. तो व्हेन वी आर सीइंग दिस लिगेंड गेटेड, और इनकी कैरेक्टरिस्टिक फीचर क्या है कि जल्दी से एकदम खुल जाते हैं, कुछ टाइम के लिए खुलेंगे. देन सेकेंड इज द आइनोट्रॉपिक रिसेप्टर. The inotropic, they will open for a particular type of ion. Then in ligand gated, they are also having the ion channels. There are three general type and each name for the congeners of glutamate for which they are responding. First is the kainate receptor. It is an acid isolated from seaweed. Then AMPA receptor, amino 3 hydroxy 5 methyl isozole 4 propanoid. So this is a full form of AMP. Sometime a question is asked, write the full form of AMP, then this. Then one is a small sodium channel and another is the channel. So the characteristic thing which you are going to receive कि कोई भी जो neurotransmitter होगा, that will be excitatory or inhibitory. It depends upon the nature of the ion channel, whether sodium is coming, potassium is going out. So which type of ion channels they are affecting, so nature will be of that type. and in ligand gated I've told you they depend. For example, I've given of G protein, then this kainate receptors, AMPA receptors, NMDA receptors. Full form of NMDA, and methyl D-aspartate, and methyl D-aspartate. Sometimes they ask write the full form of NMDA receptor, then you are going to write N-methyl D-aspartate. This is a full form. Now large molecule, you can see the large molecule transmitters, that is a neuropeptide. I told you they are big molecules. Substance P is a polypeptide and they contain at least 11 amino acid residues that is found in the intestines, peripheral nerves and many parts of the central nervous system. It is one of the family of 6 mammalian polypeptide that is called as the techinins, that differ at the amino terminals, means amino terminals will be different, but they have in common the carboxyl terminal sequence. Carboxyl terminal sequence से इनमें ये सेम रहेगा, अमीनो सीक्वेंस बढ़ता जाता है. अब one is nitric oxide, though it is gaseous but it is a neurotransmitter. The compound released by the endothelial of blood vessels, and it is also produced in the brain. It is synthesized from where? Arginine by catalyzed reaction, and in the brain in one of the 3 forms of NO synthetase. So NO synthetase, it requires NADPH. From arginine they are prepared in the presence of NO synthetase and they require NADPH. It activates guanosyl cyclase, and unlike other transmitters, and it is a gas. Which what is the characteristic feature of this gas? Because they can cross the cell membrane, so they can diffuse because they are lipid soluble, so they cross it and they enter the cell. It may be the signal by which the postsynaptic neuron communicate with the presynaptic endings in long-term potential and long-term depression. So another characteristic feature, they are lipid soluble so they can diffuse inside the cell very easily. Then there are some other chemical transmitters also, that is the purines, purines and the pyrimidines. So if we take ATP, uridine, adenosine and adenosine metabolites. So all the 3 I have told you that ADP, AMP, or they are energy currency as well, they are working as a neurotransmitters. So these purines and pyrimidines. Now coming to the neuropeptides. Neuropeptides, they are made up of 3 or more amino acid. They are large molecules and the small molecule transmitters, example endorphins and hicalins, which inhibit pain. So some are analgesic also. वो देखते ना हम एक्वा प्रेशर कराते हैं, एक्वा पंचर कराते हैं, तो उसमें क्या होगा कि पेन रिलीज़ होता है, नीडल्स को करा तो दे आर रिलीजिंग द पेन, है ना. तो उसमें भी दैट सम न्यूरो ट्रांसमीटर्स दे आर रिलीज. द न्यूरो ट्रांसमीटर एसिटाइल कोलिन. अब नाउ आई टोल्ड यू कि इट डिपेंड्स वेदर विच टाइप ऑफ आयन चैनल is opening or affecting that the neurotransmitter nature will be excitatory and inhibitory depending upon the nature of the ion channel. कि सोडियम एंटर कर रहा है तो उसका नेचर अलग होगा, पोटेशियम बाहर निकल रहा है आयन चैनल से, सो इट विल नेचर डिफरेंट है. मगर इफ़ वी टेक द एग्जाम्पल एसिटाइल कोलिन को हम देखें, एसिटाइल कोलिन है ना, इट इज़ हैविंग बोथ, इट इज़ एक्साइटरी एज वेल एज इनहिबिटरी. तो एक्साइटरी किस में है? इन द स्केलेटल मसल. कॉन्ट्रैक्शन होता है स्केलेटल मसल का, एक्साइट करेगा एकदम, कॉजिंग द मसल्स टू कॉन्ट्रैक्ट. और इनहिबिटरी किस में? हार्ट में. हार्ट की मसल्स मायोजेनिक है, एक संग उनका कॉन्ट्रैक्शन हो रहा है, लेकिन एसिटाइल कोलिन का रिलीज होगा तो क्या कर देता है? स्लो कर देता है हार्ट रेट को. तो हियर इट इज़ इनहिबिटरी. तो which this neurotransmitter is excitatory as well as inhibitory? If it is asked, then you are going to write acetylcholine. It is having both the properties. Then there are some, because of the different type of proteins they are found in the acetylcholine. So nature. Now metabotropic receptors. You have heard the name metabotropic receptors. What are metabotropic receptors? Metabotropic receptors are those, they are ion channel I told you, and these neuroreceptors bind they trigger a signaling pathway which may directly open or close the channel. मतलब एकदम सिग्नल देते हैं, जिससे चैनल या तो क्लोज हो जाएगा या बंद हो जाएगा। जैसे ग्लूटामेट एंड एस्पार्टेट, ये एक्साइटेटरी ट्रांसमीटर्स हैं। कहाँ पे? सेंट्रल नर्वस सिस्टम में। तो कौन है? ग्लूटामेट एंड एस्पार्टेट, दे आर द एक्साइटेटरी न्यूरोट्रांसमीटर्स इन द सेंट्रल नर्वस सिस्टम। GABA is the inhibitory neurotransmitter in vertebrate brain and the spinal cord। तो GABA क्या है? ये इनहिबिटरी है। ग्लाइसिन है, ये भी इनहिबिटरी न्यूरोट्रांसमीटर है स्पाइनल कॉर्ड में। तो एसिटाइल कोलिन, इट इज़ बोथ एक्साइटेटरी एंड इनहिबिटरी। ग्लूटामेट एंड एस्पार्टेट, they are excitatory transmitters, and GABA and glycine, they are inhibitory neurotransmitters. So you remember these things. It is depending upon this is the metabotropic nature. Now there are some this adrenaline, they are secreting this epinephrine, norepinephrine secreted by the adrenal medulla. They are also acting as a neurotransmitters. And how they are formed? By a protein called as the tyrosine. Another important is the dopamine and serotonin. Dopamine है ना, तो टायरोसिन, डोपामीन एंड नॉरएपिनेफ्रिन। तो डोपामीन इट इज़ वेरी इंपॉर्टेंट। क्यों? क्योंकि हमारे इमोशनल जो भी फीलिंग्स हैं, एडिटिव फीलिंग्स है, प्लेजर की फीलिंग्स है, ये किसकी वजह से आती है? डोपामीन की वजह से आती है। तो डोपामीन इट इज़ अ वेरी इंपॉर्टेंट। एंड आई टोल्ड यू न्यूरोपेप्टाइड, दे आर लार्ज मॉलिक्यूल, सम दे आर एनाल्जेसिक रिसेप्टर्स, एक्वापंचा में मैंने आपको बताया। अब यू सी दैट इन अ ब्रेन दे आर सर्टेन न्यूरो रिसेप्टर, वी हैविंग रिसेप्टर्स फॉर दिस मॉर्फिन एंड हीरोइन। अब मॉर्फिन और हीरोइन हम बाहर से ले रहे हैं, लेकिन हमारे को प्लेजर की फीलिंग हो रही है। दैट मीन्स हमारे ब्रेन के अंदर ये कोई ऐसे रिसेप्टर्स हैं इन न्यूरोट्रांसमीटर्स के लिए, कुछ ऐसी एंजॉय की फीलिंग के रिसेप्टर्स हैं, तभी तो भरे होंगे ब्रेन में कि हम वी आर टेकिंग फ्रॉम आउटसाइड, हम ड्रग्स ले रहे हैं, मॉर्फिन ले रहे हैं और वी आर फीलिंग वेरी हैप्पी। तो दैट मीन्स समथिंग इनसाइड, साइंटिस्ट ने सोचा इसका मतलब हमारी बॉडी के अंदर भी कुछ ऐसा होगा जिससे एक्साइटेटरी की फीलिंग आ जाए। जब हम बाहर से ले रहे हैं तो हमको प्लेजर फील हो रहा है। तो डोपामीन का, आप जैसे स्केलेटल मसल्स हैं, इनका कंट्रोल करना। अब पार्किंसन एक डिजीज है, उसमें आपने देखा कि उसमें क्या होता है? डीजनरेशन हो जाता है न्यूरॉन्स का, जो रिलीज़ करते हैं डोपामीन को। तो डोपामीन कम हो गया तो पार्किंसन रिलीज होगा। वी विल स्टडी इन लेटर इन नर्वस सिस्टम डिफेक्ट्स में, पार्किंसन में वी विल study in next lecture. So this Parkinson, so this is due to the degeneration of the neuron. So there are certain neurotransmitters, they are very important, and I will explain you how many type, the categories, they are depending upon inotropic, metabotropic, ligand gated, different and different type the effects. Hope you have understood, and in our next lecture we are going to study about these defects. Till then I'm asked, as usual I'm always asking a question. So what leads to Parkinson disease? And you are going to reply in the comment box. Hope you have understood. Till then, goodbye. Thank you.